This presentation is going to be from Margaret Miller, the research director at Secore International, and she's going to be presenting today on some sexually propagated corals and a little bit about coral babies in general. So take it away, Margaret.
Thanks very much, and thanks to the Coral Restoration Foundation for putting together this amazing event here virtually. So I appreciate the opportunity to talk to you today. As Alex said, primarily about coral babies. Coral babies are pretty cool and exciting, and I have the privilege to work with them a lot in my job. So I'll share a little bit about that with you today. First, just briefly, I know probably most of our audience here is very aware of the importance of corals in ecosystems because they form the foundation of the coral reef system. They provide habitat, they provide that home for so many different types of creatures. In fact, a third of total oceanic biodiversity, but they also provide us a huge amount of both aesthetic value to humans and also economic value that we enjoy. Unfortunately, the planet that we live on, we have not treated so well, and our planet is changing really fast. And unfortunately, it's changing in ways that are really bad for corals. The warming of the oceans creates these heat wave events that are very stressful to corals. They cause a lot of ill health and mortality corals when we have oceanic heat waves, which are happening more and more often with global warming. Another really devastating, catastrophic event for corals can be disease events. And I think we humans have a little bit better appreciation for the importance of these disease epidemics, perhaps than we did a few months ago. But disease is another factor that's been incredibly devastating to coral populations, especially in the Caribbean. And so these are examples of the types of catastrophes that have been happening to corals and happening at greater and greater frequencies as the environment is changing due to human activities. And so this just gives you a little bit of an illustration of what these changes look like on a coral reef. This is a coral reef in the Florida Keys. These are images from Grecian rocks, where you can see over time, back in the good old days, as we say, sort of in the 70s, that was actually maybe not even the good old days, but when we started taking photographs that we have access to, you can see such an complex landscape, seascape of branching corals and mounding corals, and pretty much everything you see in this picture is a hard coral with building that structure to a reef that we talked about. Later on here in the late 80s, most of those branching corals are dead. Some of these mounding corals are still very alive and very healthy and building a little bit of reef there. However, by the late 90s, you can see this is that same set of mounding corals is now also dead, and we see the emergence of these soft corals, which in 2004, pretty much everything that we see in the photo is a soft coral. We don't see any hard corals at all. So soft corals do create some habitat structure. They have some value to the ecosystem, but they don't build reefs in the way that hard corals do. So this is just an illustration how hard corals have sort of disappeared, in fact, from many of our coral reef environments due to these environmental changes. So as we think about the future of coral reefs, there are two really important things that I want to stress to you today. One is we need to make changes to ensure that our coastal environments present a viable habitat for corals. And right now, we're going in the opposite direction. We're making changes to the environment that are making those coastal environments non-viable for corals. So we need to fix those things that are causing those changes, like climate change and pollution and things like that. But we've really created such a big hole. We've dug ourselves in such a big environmental hole that we really need more corals in the meantime. Coral restoration provides us sort of a bridge for coral populations to be able to survive long enough for us to fix those things that are causing that unviable environment. So I'm going to talk mostly for the rest of my talk about the second half, how we're getting new corals. But I don't want you to think that the first thing that I have listed here isn't important or is less important. Both of these things are very important. And unless we fix the environment, we can plant all the corals that we want, and it's not going to make a difference in the long run. So I'll be talking about getting more corals, but changing the environment and fixing the environmental problems is of utmost importance. So how do we get more corals? Well, they actually, many coral species have a pretty amenable biology that enables us to do that. And this is just a little cartoon that shows a coral colony here, a branching coral colony, and how when a little branch of that coral colony like breaks off for whatever reason, it can form a new coral colony. And that's a really simple process. And many coral restoration activities have been very successful in leveraging this process. We call it either fragmentation or asexual propagation. And so this has been leveraged very successfully in many coral restoration operations. And many of you in our audience today may have participated in some of these operations and helped with outplanting coral fragments that have been grown in a field nursery from fragmentation. We generally use the term coral gardening for this process. And so we can accelerate the propagation of corals and place those new corals on the reef, and they can grow up. And so these are all the steps that our assistive actions can support in order to get more corals on the reef via coral gardening. And so this has been, as I said, very successful in getting a lot more coral growing on reefs in one sense, all right? And in the sense is the sort of more coral biomass. We have more colonies and more biomass of coral growing on reefs, and that's important. In another sense, we actually aren't creating more coral by this process because all these fragments are essentially clones of the coral individuals that we start with. So we're propagating a lot of coral, but it's all clonal coral. Again, it's good, it's helpful, but it doesn't create more corals in another sense. The other way that corals have, like most all organisms or animals have to get more of them is through good old-fashioned sex. And so this is a little cartoon that sort of articulates that process. It's a more complicated process than just taking a branch and growing it out. It has more steps. It involves invisible steps. Coral babies are very, very small. But it has the advantage where we are, through this process, where we have eggs and sperm from two different parents, we get genetic recombination this way. And so I have a little cartoon here that is meant to depict the process of adaptation and natural selection. So by having new genetic recombinations in these new coral babies, we provide sort of raw material for corals to adapt to these changing environments that we're creating. So there is a benefit to going through this more complicated reproductive process in enabling us to provide a little bit more raw material for adaptation to take place so that corals themselves can begin to deal with some degree of that environmental change that we're causing. So this is another approach for coral propagation and restoration. As I said, it's a little bit more complicated. There's a lot of steps involved, but through all of these steps in terms of corals spawn, most species of corals, most reef building corals spawn their gametes into the water column. So we can collect those eggs and sperm. We can perform fertilization, sort of in vitro fertilization, either on a boat or on a beach or in a lab. We can sort of husband those corals through their larval development phase. Again, all of these life phases of the coral are tiny. This picture here, you can barely see, but these little tiny dots, this is a 1 centimeter scale. Most of these life phases of the coral are microscopic, in fact. But we can still gather them together. We can provide them some assistance and protective environments and settlement substrates that can enable them to make a new home. But this is essentially the life cycle of a coral, and I'll talk a little bit more in the rest of the talk about different steps that we have developed to be able to assist all of those steps for larval propagation. The first step is collecting the eggs and sperm, and so in order to do that, we have to have some prediction or understanding when that spawn is going to happen. And so there's been a lot of historical observations that have gone into now each year we're able to create these sort of prediction calendars for different geographies. And this just shows you, this lists a bunch of different coral species that spawn or expected to spawn during different months of the year. And then the nights during each month when we expect those species to spawn with the greatest likelihood. Now, some species are more cooperative than others, but this is the first step, is to be able to predict when that spawning is going to happen. And generally, we know that the corals are using a lunar calendar that they're queuing on to be able to synchronize their spawning. So that's the first important step, and a lot of historical work has gone into being able to predict when that's going to happen. Then on the nights when coral spawning is going to occur, or we hope that it's going to occur, for most species that spawning occurs at night. And so we have night divers out. In a lot of locations, we're able to leverage and work with some of those in situ coral nurseries to be able to collect gametes from the corals that are being raised in the nurseries. But the eggs and sperm, you can see in this picture, are released in bundles and individual bundles. And those bundles will float up into the water column. We can collect them into little tents, like you see here, in a little bottle at the top of the tent as they float up. And then, as I said, we can fertilize them in a protected environment, which generally can be very successful. And then we can watch as those two coral cells become one coral cell and then starts dividing through the standard practices of embryonic development. So that's the kind of biology and those early steps that we're able to help with just by making sure that fertilization happens. Because when there are few coral parents around, that's the first step that we understand is probably failing in natural populations. Then maybe we have a ton of coral embryos. In a single spawning event, we would be able to collect hundreds of thousands or even millions of coral embryos, but they need a home to pass their larval development. So there's, again, been a lot of historic work with the idea of just trying to keep coral embryos sort of in containers, essentially in the ocean, so that they can be raised in large scales with a minimum amount of effort. And this is one of the areas that Secore, the organization that I work for, has really been investing in modernizing this approach so that we can raise these large number of coral offspring in areas where we don't necessarily have a lab. So enabled to be able to do this culture in potentially remote locations. So these are our modern apparatus. We've dubbed them coral cribs or coral rearing in situ basins, and they enable us to raise hundreds of thousands of coral babies at a time. You can see what they look like here. And then inside those cribs, we set up these settlement substrates. And that's another area where we've put a lot of effort in trying to think like a baby coral and understand what characteristics are going to help those baby corals survive. And so you can see a couple of generations of these substrates here that we've developed. They might look kind of random, but as I said, they all, if you notice, have characteristics here with grooves or pits or different microhabitats that we understand hopefully the corals can recognize. A lot of species of coral larvae like to have a little nook or a cranny where they can settle, and that can assist their survivorship after they settle. And then the overall shape, so that's the kind of microhabitats, the microfeatures on those structures. But then the overall shape of the structure also is designed in a way that they'll stabilize on the reef sort of on their own, so we don't have to paste or nail them down, and that can make it much more efficient in the process of outplanting them. So after the corals have been settled on those substrates, we can place them out in the field, and hopefully those babies grow up. And this just shows you kind of a little montage of baby corals as they grow up. This is a couple of pictures. These are Coralpalooza, the elkhorn coral, at, a couple months of age, a couple years of age. And then this is a fully grown palmata that lives in Curacao that now is spawning with their wild neighbors. So these corals can grow up, but all of these steps are difficult and complicated. And our goal really is to develop methods to improve the efficiency for all of these steps. There's a lot of steps. And in order to do that, we sort of have a three-pronged approach, similar to what I described before, where we can't wait to solve all the environmental problems before we do coral restoration. We need to do them both simultaneously. Similarly, within coral restoration, we can't wait till we have all the biology worked out and then work out kind of all the technological approaches that we want to apply that biology, and then, spread it out to help people pick up those techniques and start doing it. We really need to pursue all of these approaches at the same time because the environment's changing fast and corals don't have that long to wait for us to figure all these things out ahead of time. So Secore is involved in all of these areas of research and capacity building with the goal to make all those steps more efficient so that we can do more of it and get the number of corals out that we need to replace the ones that have been lost. So capacity building is a part of that. And we undertake training and partnership with a lot of local partners throughout the Caribbean region to implement these approaches for larval restoration and propagation and restoration throughout a wide geography. So that's most of the story of how we do sexual propagation. But again, I want to emphasize success ultimately for any coral restoration is going to depend on maintaining a viable reef environment. And that's incumbent upon all of us to figure out how to make the changes that are needed to maintain that viable environment. So I want to acknowledge the many, many partners that support Secore's work, and thank you for your attention today.